Somewhere inside your body right now, millions of red blood cells are moving through your GV-ins, carrying oxygen from your lungs to every tissue that keeps you alive. On the surface of those cells are microscopic markers that determine your blood type. Most people know the familiar categories A, B, AB, or O. Then comes the symbol that looks almost insignificant, positive or negative. But if your blood is Rh negative, that tiny minus sign represents a genetic difference that has fascinated scientists for decades. It isn't a disease, it doesn't mean your blood is weaker, and it certainly doesn't mean you're biologically separate from the rest of humanity. It means your red blood cells lack a specific surface marker called the Rhd antigen. What's fascinating is how differently this trait is distributed around the world. In many European populations, Rh-negative blood is relatively common. In East Asian populations, it is extremely rare. So why does this genetic difference appear so frequently in some populations and almost disappear in others? Where did it come from, and why has it survived for thousands of years? To understand the mystery, we have to go back to the beginning of modern blood group science. In 1940, Karl Landsteiner and Alexander Wiener were studying blood reactions involving rhesus monkeys. Their work helped identify what became known as the Rh factor. People whose red blood cells carried the D antigen were classified as Rhe positive. Those who lacked it were classified as Rh negative. What sounds like a tiny difference turned out to have enormous medical consequences. Your immune system constantly checks the cells in your body for molecular markers. When it encounters something it doesn't recognize, it can produce antibodies against it. This becomes important when an Rh negative woman carries an Rh positive baby. During pregnancy, childbirth, or certain pregnancy-related events, fetal blood can sometimes enter the mother's bloodstream. If her immune system encounters the Rhd antigen and becomes sensitized, it can produce anti-D antibodies. In a later pregnancy involving another Rh-positive fetus, those antibodies can cross the placenta and attack the baby's red blood cells. The result can be hemolytic disease of the fetus and newborn, causing severe anemia and other potentially life-threatening complications. For generations, doctors saw families experience repeated pregnancy losses or severely ill newborns without understanding the reason. The mother could appear perfectly healthy, the baby could have no obvious genetic abnormality. The problem was hidden in an interaction between two inherited blood types. Modern medicine eventually changed that. Anti-D immunoglobulin can prevent an Rh-negative mother from becoming sensitized to Rh-positive blood, dramatically reducing the risk of serious Rh incompatibility. A genetic difference that once created a terrifying medical mystery can now be managed with preventive medicine. But solving the medical problem created another question. Why did Rh-negative blood survive in the first place? If the trait can sometimes create a reproductive disadvantage, why didn't natural selection simply eliminate it? To understand that, we have to understand something about evolution. Evolution doesn't work like a machine searching for the perfect human. Genes can become common or remain rare through a combination of natural selection, migration, population size, isolation, founder effects, and simple chance. A genetic variant doesn't have to provide a dramatic advantage to survive for thousands of years. Imagine a small human population separated from surrounding groups. A few people happen to carry a particular genetic variant. Their descendants inherit it. If the community remains relatively isolated, that variant can become much more common than it was in the original population. This is called the founder effect. Genetic drift can produce a similar result, especially in small populations. The genetic history of a population can therefore create surprisingly high frequencies of traits without requiring a mysterious evolutionary advantage. And this is where geography becomes important. Rh-negative blood is much more common in populations with substantial European ancestry than in many East Asian populations. That doesn't mean every European has it, or that every Asian population has almost none. Human genetics is much more complicated than that. But the broad difference is real enough to raise questions about ancient population history. Now look toward the Western Pyrenees, where the Basque population has attracted generations of attention from historians, linguists, and geneticists. The Basques are famous for Euskara, a language isolate unrelated to the major Indo-European languages surrounding it. Their genetic history also contains evidence of deep continuity with ancient populations of Western Europe, although they were never completely isolated from later migrations and genetic mixing. During the last ice age, European populations were scattered across habitable refuges as glaciers covered enormous parts of the continent. Communities became separated by geography and climate. Some populations survived in southern regions and later expanded as the climate changed. Thousands of years of migration, isolation, mixing, and population bottlenecks shaped the genetic landscape of modern Europe. This doesn't prove that Rh-negative blood originated among the Basques, so that's an important distinction. The Basques are one population with an unusual demographic history, not a scientifically established or a negative tribe. But their history helps demonstrate how genetic traits can become concentrated in particular regions. The same principle appears throughout human genetics. A variant can be rare in one population and common in another because their ancestors experienced different migrations, bottlenecks, and patterns of isolation. That's why scientists don't look at an unusual blood type frequency and immediately conclude that there must be a hidden ancient civilization behind it. They ask a different question: what happened to the population? Where did its ancestors live? Which groups did they mix with? Which groups remained isolated? How many people survived major population bottlenecks? Which genetic variants did those survivors carry? Those questions take us much closer to the real story of Rh-negative blood. And the deeper scientists look, the more obvious it becomes that the mystery isn't necessarily that Rh-negative blood exists. The mystery is why its frequency changed so dramatically as humans moved across the planet. Now we arrive at the part of the Rh-negative story that has generated the most debate: evolution. If being Rh-negative can create a reproductive disadvantage under certain circumstances, why didn't natural selection simply remove the trait from the population? The answer begins with a misconception about how evolution works. Evolution doesn't constantly identify the best genes and eliminate everything else. It works through differences in survival and reproduction across generations, but chance, population size, migration, isolation, and environmental changes all influence which variants remain. A genetic trait can therefore survive for thousands of years without providing some extraordinary advantage. And Rh-negative blood is a perfect example of why that distinction matters. One possibility is genetic drift. In a small population, genes can become common simply because of random inheritance. Imagine a community consisting of only a few hundred people. If several of the founding families happen to carry a particular RhD variant, their descendants may inherit that variant generation after generation. If the community remains relatively isolated, the frequency can become much higher than it is in the surrounding population. Nothing supernatural has happened. No evolutionary engineer selected the trait. It simply became part of that population's genetic history. Another possibility is the founder effect, when a small group leaves a larger population and establishes a new community. The genetic composition of that founding group may be very different from the population they came from. Their descendants inherit that unusual genetic profile. This phenomenon can be seen throughout human genetics. Certain inherited conditions are unusually common in particular populations because their ancestors went through periods of isolation or were descended from relatively small found in groups. So when scientists see Rh-negative frequencies varying dramatically from one population to another, they don't automatically assume there must be a hidden biological advantage. They first ask a much more important question. What happened to the population? Where did its ancestors live? How isolated were they? Who migrated into the region? Who migrated out? Did the population experience a bottleneck? Did different groups mix? And how did all of those events change the gene pool? Those questions can explain an enormous amount of human genetic variation. But there is another possibility that scientists have investigated: natural selection. Perhaps at some point in human history, carrying certain Rh-related variants offered an advantage against something in the environment. This idea has produced several intriguing hypotheses. One of the most famous involves infectious disease. Human populations have spent most of their history fighting parasites, bacteria, viruses, and other pathogens without modern medicine. A genetic difference that slightly changed the way the immune system responded to a particular infection could potentially influence survival or reproduction over many generations. That doesn't mean Rh-negative people have a universal disease resistance advantage. They don't. But researchers have investigated whether blood group genetics can influence susceptibility to particular diseases, and this is where the story becomes easy to exaggerate. You may have heard that Rh-negative people were protected from the Black Death, or that they are naturally resistant to Toxoplasma gondii, a parasite capable of infecting humans and other animals. These claims often appear online as though scientists have already solved the mystery. They haven't. Toxoplasma is a fascinating organism. It can infect a large proportion of the world's population, and researchers have studied how genetic differences influence the effects of infection, including neurological effects. But there is no established evidence that simply being Rh-negative provides a broad protective shield against toxoplasmosis. The same caution applies to plague. The Black Death killed enormous numbers of people across Europe and Asia, and epidemics can influence human genetics by creating strong survival pressures. Scientists have found evidence that some immune-related genetic variants were shaped by historical disease outbreaks. But saying that Rh-negative blood survived specifically because it protected Europeans from the plague goes far beyond what the evidence can establish. Evolutionary history is rarely that simple. Different populations experienced different diseases. They migrated, they mixed. Some communities were isolated, some populations disappeared while others expanded. A genetic variant could survive because of natural selection, genetic drift, or a combination of both. And this is why the phrase evolution kept Rh-negative blood because it was useful should be treated cautiously. Maybe selection played a role, maybe it played a small role, maybe population history was more important. Scientists are still investigating those questions. Another popular part of the mystery involves European royalty. Medieval and early modern aristocratic families often married within relatively restricted networks to preserve wealth, political alliances, and inheritance. Repeated close relative marriage increased the likelihood of certain inherited disorders, most famously in dynasties such as the Habsburgs. But there is no solid evidence that European royal families secretly preserved Rh-negative blood as some special genetic lineage. The famous phrase blue blood doesn't mean aristocrats had different colored blood. It referred largely to the visible veins of pale-skinned aristocrats and the social idea of noble lineage. It was cultural symbolism, not evidence of a separate human bloodline. The same problem appears when people connect Rh-negative blood with red hair. Some northwestern European populations have relatively high frequencies of both red hair and Rh-negative blood, but that doesn't prove that the two traits came from one mysterious ancestral mutation. Red hair is strongly associated with variants of the MC1R gene, while RhD status involves the RhD gene and related genetic mechanisms. They are separate biological systems. Shared population history can make two unrelated genetic traits appear together more frequently without one causing the other. Then the theories become even more dramatic. Some people claim Rh-negative blood is evidence of extraterrestrial ancestry. The argument usually goes something like this. If an Rh-negative mother can develop antibodies against an Rh-positive fetus, perhaps Rh-negative people are biologically incompatible with the rest of humanity because they came from somewhere else. It sounds mysterious, but that's not how immunology works. Rh-negative and Rh-positive people are not separate species. They are not fundamentally incompatible as humans. They marry, have children, donate blood, receive transfusions, and share overwhelmingly similar genomes. The issue is a specific immune response to a specific antigen. There is no credible scientific evidence that Rh-negative blood came from extraterrestrial beings. The Atlantis theory has a similar problem. It takes unusual geographic concentrations of Rh-negative blood and connects them to the idea of a lost civilization whose survivors supposedly carried the trait to different parts of the world. But there is no genetic evidence establishing such a connection. The real explanation is less sensational but far more powerful. Mutation, migration, isolation, genetic drift, and population history. These forces can create extraordinary patterns without requiring extraordinary beings. And modern genetics has made the picture even more complicated. Not every person who tests as Rh-negative has exactly the same genetic history. In many Europeans, complete deletion of the Rhd gene is a major cause of Rhd negative status. In some African populations, different Rhd variants can produce the same phenotype. In East Asian populations, rare DEL variants can produce extremely weak expression of the D antigen. So even the phrase Rh-negative describes a phenotype that can arise through different molecular pathways. That's important because it destroys the idea that all Rh-negative people descend from one single mysterious ancestral group. There isn't one Rh-negative tribe, there isn't one secret bloodline, there isn't one ancient race. There are different populations carrying different genetic variants that produce the same broad blood group classification. And that is exactly what we'd expect from human evolution. Our species has never been genetically static. People moved, communities separated, populations expanded and contracted. Genes were carried into new environments and sometimes became common simply because of who happened to survive and reproduce. Rh-negative blood is one visible example of that enormous process, but the story doesn't end with ancient genetics, because one Rh-negative blood type has become especially important in modern emergency medicine: O-negative. Now we reach the part of the Rh-negative story that connects ancient genetics to something happening in hospitals every day: blood transfusion. When people hear O-negative, they often hear another phrase immediately afterward: universal donor. That description is useful, but it needs an important clarification. O-negative red blood cells lack A, B, and Rhd antigens, which makes them extremely useful in emergency situations when doctors don't yet know a patient's blood type. But O-negative isn't universally compatible for every transfusion, and hospitals still perform proper blood typing and compatibility testing whenever possible. Why is O-negative so valuable? Because emergencies don't wait. Imagine someone arriving at a trauma center after a serious accident. They're losing blood rapidly. Doctors need to begin treatment immediately, but complete blood group testing may take time. In appropriate emergency circumstances, O-negative red blood cells can provide an important option while the patient's blood type is being determined. But there is a problem. O-negative blood is relatively uncommon. That means blood services have to carefully manage their supplies and continually recruit donors. The same genetic combination that makes O-negative relatively rare also makes it extremely valuable during emergencies. And this creates an extraordinary connection between ancient genetics and modern medicine. Thousands of years ago, nobody knew what the Rhd antigen was. Nobody knew that a protein on the surface of a red blood cell could determine whether a transfusion was safe. Nobody knew that an Rh-negative mother could develop antibodies that might affect a future pregnancy. Today, hospitals build entire systems around those microscopic differences, and O-negative is only one combination. You can be A-negative, B-negative, AB-negative, or O-negative. The ABO and Rh systems are separate. Your final blood type is the combination of the two. Beyond ABO and Rh, scientists have identified many additional blood group systems containing hundreds of antigens. That means the familiar 8 blood types taught in school represent only a small part of the true complexity of human blood. This becomes especially important for people who have received multiple transfusions. Exposure to unfamiliar red cell antigens can cause the immune system to develop antibodies. If that happens, future transfusions may require much more detailed matching. Blood banks therefore do far more than ask, is this person O-negative? They can test for antibodies, identify unusual blood group variants, and find compatible donor units. A blood transfusion may look simple from the outside, one bag connected to a tube, blood flowing into a patient. But the biology inside it isn't simple at all. Every red blood cell carries molecular markers that the recipient's immune system can recognize. Sometimes one tiny difference can mean the difference between compatibility and a dangerous reaction. The Rh system is equally important during pregnancy. Before anti-D prevention existed, an Rh-negative mother could become sensitized after exposure to Rh-positive fetal blood. Once her immune system developed anti-D antibodies, those antibodies could remain in her circulation. A later Rh-positive pregnancy could then be at risk. Modern medicine learned how to interrupt this process. Anti-D immunoglobulin prevents sensitization in appropriate Rh-negative pregnancies. Depending on the situation and healthcare system, it may be administered during pregnancy, after delivery of an Rh-positive baby, and after certain events that could cause fetal blood to enter the mother's circulation. Modern testing can also help determine fetal Rhd status using cell-free fetal DNA from the mother's blood in appropriate settings. Think about what that means. The genetic difference hasn't disappeared, the immune system hasn't changed its basic rules. What changed is our ability to understand what is happening and intervene before the problem becomes dangerous. That's one of the most remarkable things about modern medicine. We don't always need to eliminate a genetic difference to protect people from its consequences. Sometimes we simply learn how it works, and this brings us back to the question of ancestry. If you're Rh-negative, your blood type may tell you something about the populations your ancestors belong to, but it cannot tell your entire ancestry story. A blood type isn't a complete genetic ancestry test. It doesn't prove descent from a specific ancient civilization. It doesn't prove that your ancestors came from one isolated tribe. And it certainly doesn't prove extraterrestrial ancestry. Instead, it gives scientists one small piece of a much larger puzzle. Human ancestry is made of thousands of genetic signals. People migrated across continents. They mixed with neighboring populations. They became isolated. They reunited. They experienced population bottlenecks. Some lineages disappeared, others expanded, and every generation inherited pieces of that complicated history. Rh-negative blood is therefore less like a secret identity and more like a historical fingerprint. It doesn't tell the whole story, but it tells us that human populations have never been as simple as the maps in a school textbook might suggest. And that is why the geographic distribution of Rh-negative blood remains interesting to geneticists, not because it proves something supernatural, but because it shows how strongly demographic history can influence the genes we carry. Yet there is another part of the modern story that makes Rh-negative blood even more important. Blood shortages don't care about ancient mysteries. When a patient needs compatible blood, the question isn't where the blood type came from thousands of years ago. The question is whether a matching unit is available right now. This is why blood donation is so important. Every compatible donation can become part of a chain connecting one person's biology to another person's survival. A person who has never met the recipient may unknowingly provide the blood that helps someone survive an accident, surgery, severe bleeding, childbirth complications, or another medical emergency. And because some blood types are rarer than others, the demand for certain donors can be especially high. O-negative is particularly important in emergency red cell transfusion, while other rare blood group combinations can be difficult to replace when supplies are low. Modern blood services therefore depend on maintaining a diverse donor population. Your blood type isn't just information about you. In the right circumstances, it can become an important resource for someone else. But even with all this knowledge, the evolutionary story still contains unanswered questions. Scientists can identify where Rh-negative frequencies are high and where they are low. They can identify the genetic mechanisms responsible for many Rhd-negative phenotypes. They can reconstruct ancient population movements. They can study the effects of genetic drift and natural selection, but they cannot simply look at today's map and say with certainty, this is exactly why the trait became common here. Human populations have been changing for too long and in too many different ways for the answer to be that simple, and this is where responsible science has to resist the temptation of a perfect story. The internet loves certainty. It loves statements like, scientists discovered why Rh-negative people are different, or this proves Rh-negative blood came from an ancient civilization. But real genetics is usually more complicated. Sometimes the evidence gives us a strong answer, sometimes it gives us several possibilities, and sometimes it tells us that more research is needed. The mystery isn't a weakness in the story. The mystery is the reason researchers keep investigating it. And the deeper we go, the more important one final question becomes. If science can explain the genetics, why do the alien and ancient civilization theories continue to survive? The answer isn't really hidden in the blood, it's hidden in human psychology. When we encounter something unusual that we don't immediately understand, we naturally search for a story that makes it meaningful. Rh-negative blood is unusual in its global distribution. It has real medical consequences. It has a complicated evolutionary history. Those facts are enough to create curiosity. But curiosity can easily turn into mythology when uncertainty is presented as proof. And that's exactly what happened with Rh-negative blood. The final part of this story is about separating what we genuinely know from what people simply want to believe. After everything we've uncovered, the question remains, where did Rh-negative blood actually come from? The most scientifically responsible answer is more complicated than the internet's favorite theories. There was almost certainly no single moment when an ancient or negative race appeared. The Rhd-negative phenotype is associated with genetic variation involving the Rhd gene, and different populations can carry different molecular changes that produce an Rhd-negative or weak D phenotype. Over thousands of years, those variants were carried through different populations. Some populations expanded, some contracted, some became isolated, some mixed with neighboring groups, and through all of those events, the frequency of Rh-related variants changed. That's evolution, not a perfect plan, not a straight line, not a search for biological perfection, a process shaped by mutation, inheritance, selection, migration, chance, and survival. And this explains why the real Rh-negative story is actually more fascinating than the alien story. Aliens would give us one answer. Human evolution gives us thousands of unanswered questions. Why did certain variants become common in particular populations? How did ancient population movements influence today's blood group frequencies? Did natural selection affect some Rh variants under specific environmental pressures? How did different Rhd mutations spread across different regions? And what can ancient DNA tell us about when and where these variants became common? These are questions scientists can actually investigate, and every new ancient genome provides another piece of the puzzle. Modern genetic technology has changed what researchers can reconstruct from the past. Instead of relying only on historical records, archaeologists and geneticists can compare DNA from ancient remains with modern populations. They can identify shared ancestry, population movements, genetic bottlenecks, and changes in gene frequencies that happened thousands of years ago. But there are limits. Ancient DNA is incomplete. Not every population is equally represented. And a genetic correlation doesn't automatically reveal why a trait survived. That is why responsible science sometimes ends with an honest phrase that the internet doesn't like. We don't know yet. And that isn't a failure, it's the beginning of research. The biggest mistake we can make with Rh-negative blood is taking an unanswered question and filling the gap with a dramatic story. The fact that scientists don't have every detail doesn't mean aliens are the answer. It doesn't mean Atlantis is the answer. It doesn't mean Rh-negative people are a separate branch of humanity. It means human genetic history is complicated. And complicated doesn't mean boring. Think about what this tiny difference has already revealed. A microscopic antigen helped scientists understand immune reactions. Those immune reactions helped explain mysterious pregnancy complications. The discovery led to preventive treatment that dramatically reduced severe Rh-related disease. The same blood group system now plays a major role in transfusion medicine, and its uneven distribution across populations gives geneticists another clue about the movements and isolation of our ancestors. That's an extraordinary journey for something most people only know as a plus or minus sign, and there is something deeply human about the story. Your ancestors didn't know about DNA, they didn't know about chromosomes, they didn't know about RhD, they didn't know that their children's blood could contain inherited molecular markers. They simply lived, they migrated, they survived, they had children, and they passed their genes forward, generation after generation. Eventually those genes reached you, so if your blood type is Rh-negative, you aren't carrying evidence that you're descended from aliens or a lost civilization. You're carrying evidence of something much more real, the extraordinary genetic diversity of humanity. Your blood is not a complete record of your past, but it is one tiny chapter in a story that began long before written history. And perhaps the most fascinating part is that the story isn't finished. Researchers are still studying blood group genetics. New ancient DNA discoveries continue to change our understanding of human migration. New laboratory techniques are revealing genetic variants that older blood tests couldn't distinguish, and medicine continues to find better ways to manage the consequences of blood group differences. The mysterious minus sign on your blood type, therefore, isn't a symbol of separation, it's a reminder of connection. Connection to people who lived thousands of years before you. Connection to populations that migrated across continents. Connection to families whose names and languages have disappeared. Connection to generations of humans who survived circumstances they could never have understood. And now, for the first time, we have the technology to start reading some of that history directly from our DNA. The truth about Rh-negative blood isn't that it came from somewhere beyond humanity. The truth is that humanity itself is far stranger and more complicated than we once realized. A tiny genetic variation survived through ice ages, migrations, population changes, and thousands of years of human history. Today, it influences pregnancy care, blood transfusion, emergency medicine, and genetic research. That's the real mystery. Not where the blood came from in some supernatural sense, but how something so microscopic could carry such a massive human story. So the next time you see your blood type written on a medical record, don't just see a letter and a plus or minus sign. See a clue to the enormous genetic history written inside every one of us. And if you enjoyed this documentary and want more fascinating stories about human biology, genetics, ancient history, and the mysteries hidden inside our bodies, subscribe to the channel for more.